nature. These cells divide once in approximately 24 hours, and however this distribution of cell is vary from organism to organism and also from cell type to the cell type. Through the cell 90 minutes. The cell cycle is divided M phase, and the M phase represents the phase when the actual division or mitosis occurs, and the interphase represents the phase between two successive M phases. Average duration of cell cycle. The M phase represents the phase when the actual division or mitosis occurs, and interphase represents the phase between two successive M phases. It is significant to note that in the 24 hours average duration, the cell cycles of a human cell properly last only about a hour. The interphase lasts more than 95% of division of cells. The M phase starts with nuclear division, corresponds to the separation of daughter chromosomes. Cytokinosis. The interphase, through called resting phase, is a time during which the cell is preparing for division and undergoes both cell growth and DNA replication in orderly manner. The interphase is divided into further phases. G1 phase, S phase, and G2 phase. The G1 phase corresponds to the interval between mitosis and initiation of the amount of DNA per cells occurs. If the initial amount of DNA denoted as 2C, then it increases to 4C. Even after S phase, the number of chromosomes remains 2N. In animal cells during the S phase, DNA replication S phase में कहीं प्रोफेज में कोई सिंथेसिस डुप्लीकेशन मिलता नहीं, ठीक है. In animal cells during the S phase, DNA replication begins with the nucleus and centriole duplicates the cell. So D2 phase में प्रोटीन बनता है प्रिपरेशन जो माइटोसिस के लिए. इसमें मेनली जो कंटेंट होता है साइटोप्लाज्म वगैरह वो ज़्यादा डुप्लीकेशंस होती हैं और centriole duplications. But chromosome number remains same throughout this phase. पूरे फेज में चाहे वो माइटोसिस हो चाहे वो मियोसिस हो, किसी में भी कभी भी क्रोमोसोम. एंड Z2 फेज में वही है कि प्रोटीन में ग्रोथ तो थ्रू आउट द प्रोसेस में पूरे सेल साइकिल में ये तो होती है. Some cells in adult animals do not appear to exhibit, example heart cells, exhibit divisions. तो cells at the कुछ ऐसे adult animals होते हैं जिसमें and many other cells divide only occasionally as needed to replace the cells of cells that have been lost death. These cells do not divide further and exist. G1 phase को exist करते हैं. G1 को G2 कर देगा, G1 को माइटोसिस कर देगा, ये वो कर देगा. G1 phase से exist करता है, exit करता है. G1 phase enters into inactive or quiescent stage of a cell cycle. Cells in this stage remains metabolically active. इसको inactive कर देते हैं statements में. So cell in this stage remains metabolically active but no longer proliferate unless cells do not show called on to do so. ठीक है. इस स्टेज में जाने के बाद सेल्स तब तक डिवाइड नहीं करता, डिवीजन नहीं करता. हालांकि इस स्टेज में मेटाबॉलिकली एक्टिव तो होता ही है डिपेंडिंग ऑन द रिक्वायरमेंट्स ऑफ ऑर्गेनिज्म. इन एनिमल्स माइटोसिस सेल डिवीजन is only seen in the diploid male honeybees. Against this, the plant can show meiotic division, both haploid and diploid cells. In animals, meiotic cell division is only seen in diploid somatic cells. However, In animals, mitotic cell division is only seen in the diploid somatic cells. However, their effects की जो डिवाइड करने की अलग अलग होती है. मियोसिस भी होता है, माइटोसिस भी होता है, वो एन होती है. उसमें मियोसिस तो होता है ना. मियोसिस तो पीरियड एम फेज को कह रहे हैं. दिस इज़ द मोस्ट ड्रामेटिक सारे कंपोनेंट सेल्स के, जैसे माइटोकॉन्ड्रिया हो गया, न्यूक्लियस हो गया, सारे कंपोनेंट्स को रीऑर्गेनाइज करते हैं इस स्टेज में. सिंस द नंबर ऑफ क्रोमोसोम्स इन द पेरेंटल प्रोजेनी इज सेम इट फेज इन द SNG2 phase a new DNA molecule formed are not distinct but intertwined. तो S और G2 phase में जो cells the chromosome phase. In SNG2 phase, a new DNA molecule formed are not distinct but intertwined phase. In SNG2 phase, a new DNA molecule formed are not distinct but intertwined. Prophase. The chromosome materials become untangled during the process of match together as at centromere. Centrosomes, which has under phase, begins to move towards opposite poles of cells. Prophase, which is the first stage of karyokinesis of mitosis, follows the SNG2 phase of interphase. In the SNG2 phase of new, the prophase, ये तो complex endoplasmic reticulum and nucleolus. इसका मतलब है कि सारे by this stage condensation of chromosome is completed and can be observed clearly under the microscope. Morphology of chromosome is most easily studied. At this stage, metaphase chromosome is made up of two sister